Hello everyone, I hope you're doing well. As I record this, the dregs of Hurricane Zeta are blowing outside, um, but no harm done. On the list of calamities that 2020 is responsible for, this rates as a 0. So I hope you're doing well. Um, Today we're going to talk about the term project, and again, this is for Microbial Diversity Lab. So in normal years, what we do with this term project is each student in lab isolates a variety of organisms, and they pick a couple of these, and we do a PCR to amplify a 1 kb piece of the ribosomal RNA. And these DNAs are sent off for DNA sequencing. We use a company called Eton out in the Research Triangle Park because there are a couple of folks who are graduates of our program who work there. So this year, of course, we can't do this. We can't meet together, we can't do the experiments, and we didn't isolate any organisms. But we're doing the best we can here. And so what we're going to do is that the TAs will give you a sequence. They'll either email these to you, or they may give you links to a Google Drive, something like that will take you down to a folder for each of you that contains your term project sequence. And so again, these are 1 kb sequences from the middle of the 16S ribosomal RNA. And it's from these sequences that you will do the term project. The term project is a detailed phylogenetic analysis of the organism that the sequence comes from. These projects are due November 20th at noon. Don't be late. These will be submitted electronically to the TAs. I encourage you to help each other with these. Touch bases with each other, work together on things, because there are a lot of, this is a bioinformatics intensive sort of a thing, and each of you will have questions that, maybe to someone else are trivial, but are a roadblock for you, and I don't want these roadblocks to cause problems. You, of course, end up turning in only your own work at the end. Don't wait till the last minute. Every year, I have students turning these things in the few minutes before the deadline, or worse yet, they come to me and say, Dr. Brown, the term project is coming due tonight or this afternoon, and this morning the Ribosomal Database project website is down, or Maple doesn't run on my machine, or whatever. Unless you've lived your life and never had a computer glitch, you need to make sure you leave plenty of time to get all the electronic mumbo jumbo out of the way. Maybe your internet goes down, or like I say, the Ribosomal Database project does go down once in a while, and so you're going to have to be prepared for maybe it doesn't work, or there's something that you can't figure out how to do, or the list goes on and on and on. Don't wait to the last minute to do it. In fact, my advice is, because the end of November is going to be a busy time for you guys, much better would be for you to get these things done as soon as you get the sequences from the TAs. So I'm going to move this over here. I think I can minimize it even further. No. Okay. So on the website, here's the information, the Moodle site, here's the information on the term project. And I'll reiterate, these things are due on November 20th at noon. These things are worth 70 points, so that's a pretty good chunk of your lab grade. The details of the term project are right here. There's an example term project, one that scored 100%. and the grading rubric. So there's no question about what you're being graded on here. And the TAs are going to use these to grade them, and you'll get that grading rubric turned back to you with scores on them. So let's first have a look at this one. This is a PDF. I've downloaded it, and it's over here. Here's a basic description that I've already given you. And then here are a list of things that you have to do. These are the requirements of the term projects. And notice that I've got marked in red here. This is a checklist. Each one of these items is a tangible thing that you turn in at the end of the project. Now, this is written as if you're going to print them out to physical paper and turn them in that way. In reality, since you're submitting these electronically, you're probably going to save them as electronic files, and that's fine. You can print them out and then scan them in, use a scanner app on your phone to create a PDF once you've got all the documents organized. And this is pretty straightforward, and each one of these things is a link to the description of that thing, or they're just down here, further down the page. Let's start with this a little bit. First of all is your data. The biggest difference between the project this year and normal years is, again, that you didn't isolate these organisms and you didn't do the sequencing. You don't have any of that information at all. So under microbiological information, you have nothing, and you can just say so. But you do have the other raw data, which I'm going to show you in a minute, which you should include. You've got 3 outputs from the Ribosomal Database project. You've got some phylogenetic trees, and then the write-up itself, including citations. And so we're going to go through each one of these really briefly over the next few minutes. But when you're done, when you've got everything organized, you should go back to this requirements thing and check off each of these items and make sure you have them. The easiest place to lose big points on this term project is by not turning in the stuff that we ask you to turn out. So first bit, your data. Again, you don't have any microbiological data. We're not going to tell you anything about these organisms. All you're getting is raw sequencing, which brings us to the data files. Each of you will be given access to 3 data files, and I've got the examples of them right here. And so they start out usually with a 2-digit number and a letter, 16A, 062-76-515 forward short, -F08.AB1. There are 3 that have the same name with the different suffix. AB1 is the data file from the sequencing machine. .SEQ is a plain text file with just the raw sequence letters A, G, C, and T. And then a PDF. I printed out a PDF of the data so that you can open that up and look at it no problem. Now, the AB1 files, if you wish, you can open those up with any program that you use to look at sequence data. And so I typically use, where is it? 4Peaks. Let me pull that up. .4Peaks. And so if I pull this up in 4Peaks, this example, this is what you've got. And you can scroll through this and so forth. But there's no reason to look at this AB1 file per se. I've given you a printout of this data, which is probably just as useful. And that printout is the PDF file. And so here it is. This is the data you're dealing with. For those of you who've never looked at these before, the sequence reads from the 5' to the 3' as you go across the page and down the page. The sequence call based on these dye intensities is listed at the top. This is what the text file will contain. Now you'll notice that this sequence starts off really, oh, and let me point out, the blue here is a bar graph showing you how sort of a confidence in the sequence call as it goes along. And the higher this blue bar is, the better your sequence confidence ought to be. And red here means that there's no confidence at all. Notice that this sequence starts out really bad, and then even here, you know, how many G's are these? There's a bunch of overlap in the peaks here. So this may be, is this 1A or 2? And so this isn't very reliable. Probably don't want to start with this. This looks pretty good here, but then you've got this thing here where you've got 2 peaks in the same place. You also have it here. This probably represents one of 2 things. Either this sequence was from a colony that was a mixture of 2 very closely related organisms that differ only in these places where there are mixed peaks. Or, and this is much more likely in this case, that the organism has more than one ribosomal operon, and there are tiny differences, just a nucleotide here and there, that differ in different copies of the ribosomal RNA sequence genes. This is called microheterogeneity in duplicate copies. But you can see generally the sequence is really, really nice. The confidence is good. The sequence peaks are clearly legible. For our purposes, this sequence, I mean, maybe you believe this up here and maybe you don't, but certainly by the time you get to about 70, the sequence is nice and clean and unequivocal. I would argue, looking at this, except for this one base here, this sequence is really nice and clean. These 2A's are not. And so you could start at 35 or 40 here. And as you read down here, the sequence looks really good, nice and clean. But as the DNA pieces get longer and longer, they get harder and harder to separate in the capillary columns that these things run. And you can see the peaks start to kind of run together. Now these are easy to count. This is 3 C's, 2 T's, 1 G, 1 T, 2 C's, 2 T's. But it's starting to get a little dodgy here. And so, for example, here, is this really 3 C's? Well, it sure looks like it, but is this 3 G's? Maybe 3, maybe 4. And as you can see, as you get further on, it's just a complete mess. And so there's a limit to how far you can go. Your first task is to trim the sequence to the reliable bit only. Excuse me. And so I might argue that here you see nice deflections between peaks. Here you do, here you do. But at this point, you're starting to have them run together a lot. And this is at about 640. So I might remove the first 40 nucleotides and then cut it off at about 640 or so. And you need to highlight that. That's all part of the thing described in the description. Which takes us to the next file. This is the .seq file. I'm going to pull this back over here. And this is just the data, as you can see, straight from the sequence read. And the first about 40 nucleotides of this are no good. I'm going to cut it down to these 3 G's. So if I scan along here, here they are. And so I'm going to delete those. You can open these, by the way, in any text or word processing program. It is absolutely essential, however, that when you save them as raw text. None of the analysis programs will recognize word format or anything like that. They have to be raw text. This is the most common reason for students to email me in a panic. And then I'm going to go here. I see 4 G's in a row. That's going to be easy to find. And I'm going to not quite go that far. So if I scan, these things are, I think, 80 long. So I'm looking 2, 3, 4, 5, 6, 7. As I look along here, I really ought to be able to find those 4 Gs pretty easily. Ah, there they are. Okay, so I'm going to cut them off a little before that, like so. This is my reasonable sequence, and I'm going to save this in FASTA format. And again, this is described in the write-up. Caret, right-facing caret, and then a name, like so. Caret, space, a name, a carriage return here, and then your raw sequence. The raw sequence can have spaces or carriage returns in it. It doesn't matter. And I'm going to save this then. Oh, I just saved it over the old one, which I shouldn't have done, but I save it as a new name. All right, so back to this thing. You don't have to worry about, in the examining your sequences, you don't have to worry about bad sequences or choosing one sequence over the other. You're given a sequence, and we've checked each of these sequences is a good sequence, usable sequence in this analysis. Some of them may be a little weak. Some of them may not go, 600 or 800 nucleotides long. They may only go 500. That's fine. You only need about 500 nucleotides to make this work nicely. The actual analysis is through the Ribosomal Database project, and then the visualization of the and generation of trees is through a program called Maple. We'll get to that in a minute. The RDP, the link to it is here, and I've got that up here somewhere. Here's the Ribosomal Database project. When you start at the Ribosomal Database project, you come here. I'm going to log out because I was logged in already. Have patience with the RDP. This has been around for a really long time. It sits on an old server on somebody's office desk. It's not particularly fast, and it's not particularly user-friendly. The not being fast is something that you will find annoying, but get used to it, I'm afraid. The first thing you have to do is log in, and when you log in, you just run it as a test drive. We don't need to keep our sequences private. Notice that it's taken a while here to load the next page. That's par for the course. Okay, what they're showing you here under Overview is a list of sequences that have been submitted to the database. And so the plus sign here means that if you click on that, it will select it. It's got a green plus or a greenish plus. That means it is not selected. It's got a name, who the submitter was, which is almost always going to be RDP demo because that's the user account you're using. The date, the project, you can put a name on the project, what the gene name is, and then this column is for aligned, failed, or unaligned. And so the number here will tell you how many aligned sequences there are. You can see they don't always work. They get stuck, and there it is. So you need to upload your sequence. So I'm going to upload. This is a bacterial 16S ribosomal RNA. Assign a name, MB452 test. Project. I'll give you a description for what you should put in here just so you can keep track of it in the notes. I'm going to choose a file to upload. Go to my desktop, pull this thing up. And upload it. It says upload sequence group contains 1 sequences. You can upload FASTA files with almost any number of sequences in it. We're doing them one at a time. But what this means is that it recognizes one sequence in that, which means you formatted the file correctly. Click Continue, and it will take you back to the overview. There you go. And you can see here's that sequence right there. I'm going to select it. This is another place where you can go wrong. If you're going to do analysis on your stuff, you have to have it selected. And again, this takes a while. And you'll notice that this green plus turns into a red minus, meaning it's selected. And if you click on it, there you go, that will deselect it. There are a couple of analyses you have to run. I'm going to go through the first two because they're really easy. The first maybe is the classifier. And so you've got one sequence selected. That's the one I want to run it on. I'm going to run it on the standard 16S RNA training set. I don't need to choose a file or paste it because I've already got it selected. So do classification with selected sequence. And this is going to run it through the sequences and bin it, tell you what kind of an organism it thinks it is. And so here we go. It thinks it's a Pseudomonas. First of all, it's sure that it's 100% sure that it's a bacterium, Proteobacterium, gamma Proteobacterium, from the order Pseudomonas, from the family Pseudomonaceae, and the genus Pseudomonas. And if you click on that, it will tell you show you the hierarchy. And so you've got a Pseudomonas. And it's 100% sure of that. The next thing I'm going to do is a sequence match. All this does is find the closest matches in this database of millions of sequences. Now, you don't want it to search everything. It is critically important that you constrain this search to certain kinds of sequences. So we're going to look for type strains only. This way you should only have one sequence from each characterized species of organism. It doesn't always work out that way, but it's pretty close. You're only looking at isolates, meaning you're not looking for sequences extracted out of environments. Knowing that your organism is very similar to environmental sequence 1342-9 doesn't tell you anything. You don't know anything about that. Unless it's a named, isolated organism, you don't really know anything about it. We're going to make sure that it's searching for full-length good sequences. And we're going to ask it to send the 20 best sequences back. Do sequence match with selected matches. And so here we go. Again, it's showing a bunch of stuff from Pseudomonas. So let's take a look. View selectable matches. And so this purple column is fractional similarity. And so 1.0, that means the sequence you submitted is identical to that of Pseudomonas corrugata. It is also identical to those two species, this one, and it's 99 point something similar to all of them. This is the issue with getting a Pseudomonas, right? Is that there's a bunch of species and they're all very, very closely related. The second number here is an SAB value, which is related to similarity, but it's weighted based on conserved versus non-conserved sequences. Now, any one of these, so for example, if you wanted to say, okay, if this looks like corrugata, what is that sequence? And you can click on that, and it will show you the sequence record for that, and you can look up information about it. You can also select them so that they're included in any future analysis you do, and in particular, any phylogenetic analysis. As you're going through these things, Here's the sequence match description. Here's the classifier. Remember to save printouts, either electronic printouts or physical printouts of all the right stuff so that you have it. The next part of the analysis is the construction of trees. And for this process, you need two things. You need the RDP. This is where you're going to get your collection of sequences to do treeing from. And you need Maple to run the actual treeing and visualize the trees and manipulate the trees. And the links for those are here. And the process for all this is described in great detail here. And I'm not going to go through it. I expect you to figure all that out. But I am going to show you how to use the browser and the RDP. So if I go to the browser, this is a taxonomic browser. And you can see it starts at the deepest level, the root. Notice that the selection icon here is split. It's part red and part green. And that means that some sequences are selected and some aren't. Here's the domain bacteria, and some are selected and some aren't. And if you look at these different phyla, you'll see that when we get to Proteobacteria, some of those are selected and some aren't, because this is where the Pseudomonads are. You've also got archaeal and some fungal sequences in here. We've got this set still to only look at type strains and isolates and good sequences so that we're not looking at just environmental stuff. And so if I want to select sequences related to it, I'll go here to Proteobacteria, and that breaks open the Proteobacteria and showing, for example, the alpha proteos, the beta proteos, the deltas, etc. And here are the gamma Proteobacteria and the Pseudomonad ales within there. And so I'll click on that. I've got some of the family Pseudomonads selected, and everything that's selected is in this genus Pseudomonas. And so there's 4 pages of Pseudomonas species, and this tells you the ones that are selected. Those were the ones selected from the sequence match. Now, if I want to add a sequence, let's say, for example, okay, I've never heard of Pseudomonas Veroni, but it was one of the close matches, so that's good. These are all the close matches. But I like to include a Pseudomonas in there that I'm familiar with. Well, I know Pseudomonas fluorescens. I'll select that one and add it to the list. So when I generate a tree, you'll have Pseudomonas fluorescens in the tree. As you scan down here, maybe there's another Pseudomonas you're familiar with. Most of you are familiar with Pseudomonas aeruginosa, so that's what I'm looking for. There's Pseudomonas syringae. Maybe you're familiar with that. Maybe, oh, we'll select it. Pseudomonas syringae. There's Pseudomonas aeruginosa, selected. All right. So I've got a bunch of Pseudomonas selected. But if you're going to generate a tree, you might want to include some things that aren't in this genus Pseudomonas. And the best way to do that would be to zoom out and collect some sequences from its relatives. And so maybe I'll do an Azomonas. They only have 2. I'll pick Macrocytogenes because I happen to be familiar with it. Azorhizophilus, you'll notice that there aren't any sequences there. These are the number of selected. The first number is the number of selected sequences. The second one is the number of sequences that fulfill these constraints. And the last one, I don't remember what that means. But the point is that there are no type strains of Pseudomonas, of Azorhizophilus, so we won't include that. I'm sorry, my lights seem to be blinking out. Here's an Azotobacter. I'll pick that one. Vinlandii is the most commonly known or familiar of those. Just for fun, I mean, there's only 2 of those and 6 of those, so I'm just going to select them all. All the Azomonads, all of the Azotobacters. There aren't any serpents. There's only one Permeabacter, whatever that is. There's a Thiopseudomonas and an Oblitomonas. Okay. And if I click on that, you'll see that they're both it's selected. Now this unclassified Pseudomonas, what are those? Here's Azorhizophilus paspali, don't know what to do that. Pseudomonas thermotolerans, sure, why not? And I can go back if I want to even a little bit further. So here's the order Pseudomonadales. I've got these things in the family Pseudomonadaceae selected. But maybe I ought to put some other representatives a little further out. So here's a Ventosomonas. I'm just going to select both of those. Here's an Acinetobacter. I'm not going to select all those. There's 38 sequences. So I'm going to just click on that and pick one. Now I don't know that much about Acinetobacter. The only one I've ever heard of, I've heard of Biorhynchi, and there's one called Calcoaceticus. I don't see that right off. Well, here's Baumannii, so I'm going to select that one. You could basically select them arbitrarily. Go back to Pseudomonadales. So I've got an Acinetobacter selected. Maybe you've heard of Moraxella before. Moraxella cataralis causes pink eye. I'll select that. Psychrobacter. Yeah, why not? Frigidicola is the only one I've ever heard of there. And you can decide for yourself. Those are all things from the family Moraxella. Maybe we'll want to back up a little bit again. Gamma proteobacteria are a big group. Maybe people aren't familiar with these organisms. But if you know anything about bacteria, you know about E. coli. So I'm going to back up here. Here's Enterobacteria, Escherichia shigella, Escherichia coli. And so what you're doing here is you're selecting sequences based on what you think might make a good tree. And so is E. coli a good enough outgroup? Or I mean, maybe you want to include it in the general proteobacterial scheme. And so maybe you've got E. coli, that's one Enterobacterium. Maybe you want to take one Chromatium, and so you can choose a Chromatium. Whoops, let's go to Gamma proteobacteria. Choose one Alteromonas. And notice I'm picking the genera that have the same name as the orders. That way it's easier to pick through them. I don't know anything about these organisms, so I'm going to pick one arbitrarily. We've got an enteric, Cardiobacter, Cardiobacterium hominis. That sounds like a human pathogen. Legionella. Surely you've heard of Legionella before. Here's Legionella pneumophila is the main one. There you go. And you keep doing this until you think you're done. You can pick as many of these or as few of these as you think best. Once you've got all the sequences you want to try selected, you're going to download those sequences. FASTA format. You can download them either aligned or underlined. It really doesn't matter, and the reason for this is that Mabel is going to realign them anyway. And I'm not going to go through the process at this point. You can see you've got 46 sequences. One thing you want to be absolutely clear about, and maybe you want, before you download, to go to the overview. Make sure your sequence is aligned. Here we go, my RDP sequence. So your sequence is in there, so select it still, so that's good. Download them, and then go to the Maple website and generate some trees and manipulate them, all as described here. It's going to be an iterative process. You're going to get a tree that maybe looks something like this. You're going to look at this and decide, yeah, I don't need 5 Bosia sequences in this tree. One representative makes the point. And so you're going to go back to the RDP, remove all but one of them. Maybe you need to add some more Pseudomonas, whatever. Go back and forth until you're happy with the most informative tree you've got. In Mabel, you can rearrange the tree, flip branches, turn things around, draw them in different formats, all that kind of stuff. And so in this example that's done here, it's nice, for example, to have your sequence at the very top and the outgroup here at the very bottom. Once you're happy with the tree, then you need to annotate it, and that's going to appear probably as a figure in the final report, but also your primary and final trees and so forth are all part of the things that you have to turn in. Speaking of the write-up, once you've generated all your data, once you've done all your analysis, the final thing is to write it up. All of your data is going to be attached to this as accessory data, but the paper itself is going to be in the format of a real scientific paper. This is limited to 2 pages, single spaced. You can format this. It's nice to have it formatted, double column, as if you were going to actually publish it. And it's the normal parts of a scientific paper: title, authors, institution, a 2 or 3 sentence abstract, an introduction, materials and methods. These can be brief. And then your results, including probably a figure, and that figure would seem natural to be your final annotated tree, and a discussion. You need also, of course, to include citations. I've given you an example of this here. This is one from 2017 that the student turned in. And as you can see, it looks like a real scientific paper. Once again, the limit is 2 pages for the paper itself. And then attached to that is all the accessory data. There are some preliminary trees. This student had a Burkhold area. These are all the things I've asked for. This is the sequence itself. Once you think you've got this thing completed, make sure, once again, that you go back to that checklist and then have a look at the grading rubric and make sure that you're going to get top score on all these subcomponents. All right? Okay, so that's the term project. Oh, here's the Maple website. And you just do one-click trees. That's really the way to do it. Upload your alignment from the RDP. Okay, so that's it for the term project. Let me remind you once again that these things are due on November 20th at 12 noon. They need to be in the email outbox to your TA by that time. You can actually send them to either TA, and they're sorting out how to do the grading between the 2 of them. If you have any questions about this, you can feel free to drop me an email. I'm happy to give whatever help is necessary in order for you to do a good job with these. Likewise, once again, feel entirely free to work together with your classmates on this. Realizing, of course, that you have a unique sequence, and all of the things that you turn in are going to have to be your own work. All right, thanks so much. I hope you have fun with this, or at least have an informative time of it. Take care and stay safe. Goodbye.